Morning, everyone. So, on last class, we discussed about the digestion of carbohydrate. So, before going to digestions of carbohydrate, we should know about what are the dietary carbohydrate. So, dietary carbohydrates means those carbohydrates which we include in our daily food. So generally, when talking about these different carbohydrates which we include in a dietary food, there's one starch, so it's the polysaccharide, and then sucrose, which is the disaccharide, the lactose, so which is again disaccharide and present in a milk, and then these monosaccharides like fructose, glucose. So even we are taking this polysaccharide, disaccharide, and monosaccharide. So before they absorb in our body, they should be converted into the monosaccharide. So that means starch should be converted to glucose. This sucrose should be converted to fructose and glucose. So lactose should be converted to galactose and glucose. So, and these conversions of these disaccharides, polysaccharides into the monosaccharide is a digestion. So that is the hydrolysis. So that is converting to the monosaccharide. And in our body, these conversions is done by enzymes. So, these those enzymes which catalyze these polysaccharides or disaccharides are considered as they are the enzymes. So it is the enzymes which is there are different enzymes are present in our digestive system, which is participate in a digestion of carbohydrate. On that we have basically two groups of enzymes are there. So one is that is the amylase groups. So these amylases, so amylases, and next one is the this disaccharides. So amylases are amylases. So we have two types of amylases. One is salivary amylase, and another one is a pancreatic amylase. And this salivary amylase and pancreatic amylase. So these are the two isoenzymes. So they cleave the alpha 1,4 glycosidic linkage presents in a amylose. So while cleaving this alpha 1,4 glycosidic bond present in a starch, or that means in amylose. So what they do means they clips this alpha 1,4 linkage randomly. So that's why what happened, this amylose or this polysaccharide that is converted to the oligosaccharides. So certain of these oligosaccharides, and then these disaccharides, and sometimes some few monosaccharides also. And this salivary, while we are talking about this salivary amylase, so the salivary amylase, it is present in saliva, and for the activity of this amylase, it should require this neutral pH. Uh, so, due to the short interactions with the food of this salivary amylase, uh, as the actions of activity of this salivary amylase is insignificant, because this salivary amylase, as soon as it reached in the stomach, it become inactivate. So the major amylase which completely hydrolyzed is amylose presence in a starch or glycogen. So that is a pancreatic alpha amylase. So pancreatic alpha amylase, it acts on the intestine, and it cleaves the remaining this starch, so which is not act by this salivary amylase, as well as this short oligosaccharides, so which is the degraded products of this degraded product of starch that is cleaved by a salivary alpha amylase. So that is cleaved by a pancreatic alpha amylase. So the remaining, so the degraded product is that is a short this one oligosaccharides, disaccharides, and these oligosaccharides and disaccharides that is cleaved by the another enzymes called as disaccharide enzyme. So that is a maltase, isomaltase. So they clips these oligosaccharides and disaccharides into the monosaccharides. So finally, what happened? All these disaccharide, polysaccharide, which is present in dietary carbohydrate, they are converted into the free monosaccharides, and this only these free monosaccharides can be absorbed from a epithelial cell of from a this lumen of intestinal lumen of small intestine. So, I'm talking about this while talking about this absorptions of these monosaccharides. So basically, these monosaccharides from this lumen of small intestine to the epithelial cell of small intestine by two mechanisms. So one by means of a secondary active transport. So basically, it is glucose and galactose. They use this route of transmission, and next by the facilitated transport. So that is basically that is used by a fructose and mannose. So while talking about this absorptions of glucose and galactose from a lumen of intestine, so there is a specific carrier protein called as a S-GLUT. So S-GLUT means that is a sodium-dependent glucose transporter. And generally, while talking about this sodium glucose transporter protein, so it is found in the two sites in our body. So one is in the intestine, and next one in a renal tubules. So this S-GLUT, sodium-dependent glucose transporter, which is present in the intestinal lumen, that is also called as a S-GLUT1. And next one is sodium glucose transporter, which is present in the renal tubules cells, so that is called as a S-GLUT2. So, this for the mechanism of how this glucose and galactose transport across the this apical side of a epithelial cell from an intestinal lumen. So for that, what happened is the concentrations of a sodium is higher in intestinal lumen than a intestinal cell. So the movement of the sodium ion is towards the down toward the concentration gradient. So that means higher concentrations to the lower concentrations. So this when this sodium bind to its carrier protein, it changes the conformations of this S-GLUT protein. So that what happen, it also this glucose and galactose they can also bind. So along after the binding of glucose and galactose and sodium and that transport protein, then what happened? These both are carried towards inside the epithelial cell. And during the transport, what happened is glucose and galactose. It is transport against a concentration gradient. So that means the concentrations of glucose and galactose is higher in intestinal lumen and lower in epithelial cell. So this transport is against the concentration gradient. And for the any chemical constituents which travel against a concentration gradient, they require energy. So here also what happened for the transport of glucose and galactose from lower concentration, that is the intestinal lumen, to the epithelial cell, that is in a higher concentration. So for that it require energy, and the energy which is necessary to transport from lower concentration to the higher concentration, it is provided by a energy which is transported from higher concentration to the lower concentration. So that energy is provided by a sodium. After that, what happened? So after reached to the epithelial cell, the sodium again it transport back to the interstitial space with exchange of potassium. And here what happened? It moved from lower concentration to the higher concentration. So in this time, what happened? It requires energy, and this energy is provided by a ATP, and this transport of sodium from epithelial cell to the interstitial space. So that is carried out by the ATPase enzyme. So for the again back of this sodium from epithelial cell to the interstitial space is for that it required energy ATP and the another enzyme ATPase. So here what happened? The energy necessary for the transport of glucose and galactose from intestinal lumen to the epithelial cell. So that is it 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 require energy, but it do not consume a direct energy, but it consume energy indirectly. So this type of transportation, so this type of transport, so it is called as a secondary active transport. So generally active transport, when we are talking about the active transport means that is the requirement of the energy. So if it directly consume energy, so here this transport of sodium from epithelial cell to the interstitial space. So here what happen, energy is consumed directly. So this type of transport is called as a primary active transport, and this type of transport mechanism is called as a secondary active transport. Here energy is consumed indirectly. And next system, that is facilitated transport system. For this facilitated transport system, here different carrier proteins are there. This glucose, these are called as a glucose transporter. And while talking about the mechanism of this glucose transporter, so generally these carrier proteins, so they can exist in a two form. So one is PONG form, next one is a ping form, ping conformation. So ping conformation, PONG conformation. So generally, these type of transporter, this type of transporters are bidirectionals. So they are bidirectional transporters, and generally these ping and PONG conformations. So these PONG conformations is then always it is towards the higher concentrations. So that means, so here what happened is fructose and mannose. So they are higher concentrations. So that what happened here, this transporter protein, it exists in a PONG state, PONG conformation. So during a PONG conformation, so what happened now, the molecules, these molecules get bind. So after binding of these molecules on a PONG conformations, now this protein, it changes its conformations to the ping conformations. And these ping conformations, then what happened, it releases the molecules to the cells. So when it's become a empty, then again it convert to the PONG state or PONG conformation. So it moved toward the higher concentration. So that again another molecules can bind with this transport protein. So in this way, what happened? This facilitated transport, that this transporter carrier proteins, they work. So up to here we discuss on last class. So ping pong mechanism. So the carrier proteins exist in two conformations depending on the solid concentration. So pong state and ping state. So in the pong state, it is exposed to high concentrations of solids, and molecules of solids bind to specific sites on the carrier protein. So in the inner site, a conformational change occurs to ping state, and the solid is discharged to the site favoring equilibrium. So the empty carrier protein then reverts to the original conformation PONG state to complete the cycle. So here you can see on the figure. So this is the initial, that is the PONG state. So that glucose get bind. So after the binding of glucose on that PONG state, now it changes its conformation to the ping state. So now what happened, from ping state, that glucose gets released. So after the glucose get released from this ping state, now this carrier protein, it changes its conformations to the PONG state. So it's complete the cycle, PONG state, and then again another molecule of glucose can get bind over there. So while talking about this glucose transporter, so we have a different types of glucose transporter. So from glucose transporter 1 to 7 in a different sites in our body, and they are involved in a transport of glucose to the various cells. So you can see here. So GLUT1, so especially it is present in a red blood cells, brain, kidney, colon, placenta, and it is a glucose uptake by these cells independent of a insulin. And generally it is GLUT2 1. So they can able to transport the glucose even in a low concentration. So they have a high affinity. So that means high affinity means that these red blood cells, they are able to get glucose even the persons is in a hypoglycemia. So that is a GLUT1. So next one is GLUT2. So it is generally present in a liver, kidney, small intestine, beta cells of pancreas, and it have a low affinity. So low affinity means that this glucose transporter, this GLUT2, only open when the concentrations of glucose is high. And the specificity for this GLUT2 is that it helps in a rapid uptake or release of glucose. So generally, during a fed time, this fed state. So that means after consumption of a food, then what happen? The food get digested and they are absorbed. So that what happen? There is a increased level of glucose in a blood. So that time what happened? This from blood, this liver, this kidney, so they and these beta cells of pancreas they uptake this glucose via glucose this glucose transporter 2. So and when the person's in a fasting state, so that time what happened? This glucose will be closed, so that this liver, kidney, small intestine, this they cannot able to uptake a glucose, or they cannot supply the glucose. So GLUT3. So this is the another glucose transporter. So generally presents in a brain, kidney, and placenta. So, and it is work as a glucose uptake. So next one, the important one is this GLUT4. So glucose transporter 4. It is generally present in a skeletal muscle, adipose tissue, and heart. And for the activity of this glucose transporter 4, it depends upon a insulin. So when insulin is present, so that time what happen? This insulin get bind on the glucose transporter 4, so that this will be open and they are able to transport a glucose. And this GLUT4. It has a very clinical significance, and generally due to deficiency of this glucose transporter 4, or due to the deficiency of the insulin, then what happen? The persons get a diabetes mellitus type 2. So in case of diabetes mellitus type 2, then what happened? Due to the lack of this glucose transporter receptor, this glucose transporter or this insulin, then what happened? Though the concentrations of glucose is high in a blood, these skeletal muscles, adipose tissues are not able to uptake that glucose for its utilization. So because of that, what happened? The person is in a hyperglycemic condition. So next one, that is the GLUT5. So this is the glucose transporter 5, and so though it is a name, it is a transporter of glucose, but it is more specific for a transport of other carbohydrate. So that means it is more, this glucose transporter 5 is mainly specific for a transport of fructose. So that it is present in a majorly where the fructose is necessary, or where the fructose is transported. So such as that in small intestine, testes, sperm. So next one, GLUT7. So it is a glucose transporter 7. So it is present in a liver and endoplasmic reticulum, and its main function is that the transport of glucose from endoplasmic reticulum to the cytoplasm. So in our body, apart from these two mechanisms, that is the facilitated diffusion and then this one active transport, the another mean of transport is also available. So which is called as a paracellular route. And these the two method which we discussed earlier, so that is the facilitated diffusion and active transfer, they are called as a transcellular route. So that means across the cell. And here this another means, so it is called as a paracellular route, and that is between the adjacent enterocytes. So water can enter the intestinal space through this route and take with it small molecules including glucose, amino acids, and small peptides. And this paracellular route is also known as a solvent tract. So you can see in a figure via this from intestinal lumen to the this blood via the cell, that epithelial cell, that is called as a transcellular absorptions. And so in between these two cells, there is a small space is there, and from that space, if the transportation takes place, then it is called as a paracellular absorptions. And this type of absorption, it is also called as a simple diffusion. Now, clinical significance or clinical disorders regarding to the digestion and absorptions of carbohydrate. So most common this disorder of this carbohydrate digestions and absorption is that is a lactose intolerance. So lactose intolerance is a clinical conditions of pain, nausea, and flatulence after the ingestion of a food containing lactose. So most notably dairy products. So when those persons who are lactose intolerant, so if these people they consume a milk, that means a dairy product, then they will have a symptoms like pain, nausea, and flatulence. And the main cause for a lactose intolerance is lactase deficiency. and is lactase enzyme. So that is responsible for a conversions of lactose into the glucose and galactose. So when this lactase enzyme is deficient, then what happens? This lactose cannot be converted into the glucose and galactose. So that what happens? When we consume this lactose containing foods, then it is not able to digest. So types. So classified generally this lactose intolerance. It can be classified into three types. So one is primary severe lactase deficiency. Primary severe lactase deficiency. Next one is primary low lactase. So primary low lactase deficiency. And third one is secondary low lactase deficiency. So primary severe lactase deficiency means here there is a deficiency of lactase during a burn. So that means as soon as the burn of the baby, so he or she is deficient from a lactase enzyme. So it is a inherited deficiency of lactase. So generally it is seen after a birth of a baby. So in that case, what happened? After when the infant is feeding with this breastfeeding, then what happened? This infant he or she get a this one diarrhea and this abdominal cramp and this flatulence. So this is the symptom of a lactase intolerance. So another one is so it is seen as soon after a birth of a child or birth of an infant. So next one is that is the primary low lactase deficiency. So in this case, what happened? In this case, lactase intolerance is not occur in the initial phase. So at the later phases, suddenly there is a decline of this lactase activity in the person. So that what happened? This person is difficult to digest lactose. And generally, this type of this primary low lactase deficiency, it is common. So it is the most common case of lactose intolerance, and it is mostly present in these non-white populations of America, and even it is also seen in case of these Southeast people. So like in India, so generally it is seen. Generally, it is seen in the elderly people. So there is a gradually decrease in a lactase activity, so that the person is not able to tolerate the lactose. So next one is that is the secondary low lactase deficiency. So in this case, what happened? There is a lactose intolerance is occur due to the another causes. So that means there is the different another this secondary disease. So like this, so intestinal disease such as celiac disease and gastroenteritis. So on that case, what happened? This lactose is not able to digest. So clinical feature, the clinical feature for this lactose intolerance is diarrhea, flatulence, abdominal cramp, and distension. Distension. So that is abdominal discomfort. So then this lactose. So here, due to the deficiency of lactase enzymes, what happens? Lactose cannot be digested. So that means it cannot be degraded into the glucose and galactose. So that's why what happened is lactose is it move small intestine without this one catalyze, and so because of that what happens is lactose it has a osmotic effect, so that it drags the water from a peripheral cells, so which increases the water content of a water content of a stool, so lead to the diarrhea. And next, this lactose is also so lactose is so it is it reaches to the large intestine, and where this lactose they are fermented by a bacteria which is present in a intestine, so that it is converted to the different this one gases like this hydrogen, methane, and carbon dioxide, so which lead to the flatulence, abdominal cramp, and distension. So diarrhea is due to the osmotic effects, so that is the water drag from a small intestine. from these epithelial cells to the intestinal lumen, and this flatulence, abdominal cramp, and distension is due to the this productions of gas after fermentations done by the intestinal bacteria. So these are the clinical feature of a lactose intolerance. So management. So for the managing of this lactose intolerance, so main management is that is avoiding the intake of milk. So milk contain lactose. If we avoid lactose containing foods, then this problem will not take place. So next we can use of a live lactobacilli. So lactobacilli, they have a lactase like activities. So they also contain the lactase enzyme, so which helps to digest this lactose into the glucose and galactose. And here use of yogurt. So that is also the similar. Yogurt contains the lactobacillus, so which have a activity of a lactase, and that lactase can hydrolyze lactose into the glucose and galactose. So another this deficiency is a sucrase deficiency. So this is the very rare disorders, inherited deficiency. So here where this sucrase is deficient, and most of the time this, when the person is having a sucrase deficiency, that time they also may have a isomaltase deficiency, because this sucrase and isomaltase, the both enzymes, so they are synthesized during a synthesis, they are synthesized in a single polypeptide chain. So this sucrase and isomaltase, they are a single polypeptide chain having a two different active site, one for sucrase and one for isomaltase. And those persons who are deficient on this sucrase and isomaltase, then they are unable to digest sucrose and isomaltose, and the symptoms are same like lactose intolerance. So that is a diarrhea, there is abdominal cramp, distension, and flatulence. So another deficiency, that is the monosaccharide malabsorptions, and this is also a rare disorder, and where this carrier protein, so like this S group, sodium dependent glucose transporter 1, so that is the deficient on this case, so that this glucose and galactose, they cannot be absorbed. And in this case, what happened, there is a hypoglycemia is present, so due to the unable to absorb these monosaccharides. So now next, that's dietary fibers. So before starting these dietary fibers, we should know about this one dietary carbohydrates. So this dietary carbohydrates in a single word. So that is a carbohydrate presence in diet, and those carbohydrates presence in a diet that we can simply distinguish, that can be classified into two types. One is digestible carbohydrate. So next one is non-digestible carbohydrate. So digestible carbohydrate, which can be acted by the enzyme presence in a GI system. So that is called as a dietary carbohydrate. And next part is that non-digested carbohydrate, so which is, these are the carbohydrate which is not digested by our body. So these carbohydrates, so those non-digested carbohydrate are called as a dietary fiber. In a single word, these dietary fibers are a non-digested carbohydrate. And this for being a dietary fiber, it should be present in a edible plant food. So these dietary fibers are found in a plant food, so edible plant foods. And so these fibers, they can be complete or partial fermented in a large intestine. So dietary, we are talking on this dietary fiber. So dietary fiber, on simple words, so it is a undigested carbohydrate. So these are present in a food which is not digested by our body, so which is called as a dietary fiber. So being a dietary fiber, it should be from a plant source, and which are edible, and it should be complete or partial fermented in large intestine by a bacteria. So generally, these dietary fibers, due to the lack of the enzymes in our body, they transport from mouth to the direct to the large intestine. So there is a no activity on these carbohydrates in between these parts. So that means this from mouth to small intestine, there is a no activity on these carbohydrates, because the body do not contain enzymes which is able to hydrolyze these carbohydrates. So they are directly transferred to the large intestine. However, in large intestine, what happened? There is a presence of bacteria. So this bacteria is able to hydrolyze this carbohydrate. So depending upon some dietary fibers, they are completely fermented or completely digested, or some they are partially digested in a large intestine by a bacteria. So these dietary fibers promotes beneficial physiological effects including laxation and or a blood cholesterol attenuation and or blood glucose attenuation. So these dietary fibers, they prevents from constipation, they prevents the increasing of blood cholesterol and blood glucose. Classifications of this dietary fiber. So classifications of dietary fiber on the basis of these physical properties. So that means on the basis of physical properties, these dietary fibers can be grouped into the three groups. So one is soluble. Soluble means those dietary fibers which are water soluble. So example of these soluble dietary fibers are pectins, gums, mucilages. So next one, that's the insoluble to water. So the example for insoluble dietary fiber is cellulose, hemicellulose, and lignin. And next one is resistant starch. So this resistant starch is, that is the starch only which are not digested. And the example for this resistant starch is that we can give that while the cooked and cold starch from a potato and rice. So this type of this resistant starch, that is also used in a weight losing program also. So for that, for making a starch to a resistant to digest, so generally what it is done is that preparing for this resistant starch is, first the rice or this rice get cooked, and after this well cooked rice, then it is kept in a refrigerator. That means to keep it is kept in a cold temperature. So and later on it is consumed. So that means by heating it is consumed. So on that time what happened, the starch become a resistant, so which is not digested, and then it's become a dietary fiber. And generally this type of starch is generally used for a this weight losing program. Okay, so now we'll move on to the this one different this parts. So soluble fiber, so these are dissolved in water and form a gel like substance. So these soluble fibers are generally found in foods like fruits, oats, beans, and barley. So these soluble fibers are more helpful in support the growth of friendly bacteria, and it reduces the cholesterol absorption. So it binds to the dietary cholesterol. The soluble fibers, they inhibits the absorptions of cholesterol from a small intestine. Apart from that, it also increases the excretion of bile salts. So that what happened, these soluble fibers, they inhibit the this reuptake of bile from this one large intestine. And so that what happened is the cholesterol which is present in a body, that is converted to the bile, and so that it enhances the excretions of a cholesterol, so which make a reduced level of a cholesterol in a body. So these soluble fibers, they helps in a decreasing cholesterol level. So and next, this beneficiary of soluble fiber, it slow down the time for food to pass through stomach into the small intestine. So which makes slow absorptions of a glucose. So which so that there it prevents a sharp rise of a blood glucose after a food consumption. So that is the, it decreases the postprandial increase of glucose. And next one, it's give a satiety for a long time. So that means it helps to it help the fullness of a stomach for a long time. So that means it decreases the appetite of a person. So insoluble fiber. So insoluble fibers are those fibers which does not dissolve in water, and generally these insoluble fibers are found in the foods like a wholemeal bread, wheat bran, that is vegetables and nuts. And these insoluble fibers, they are necessary for making a bulk stools, so that it prevents the constipation. Resistant starch. So it is, as discussed, so it is developed by heating and cooling of some foods such as potato and rice. So few grains or grain products have been developed for their high resistant starch levels. So example as is high maize and barley max TM. So these are generally these artificial made foods. So on which these starch made resistant from digestion. And the significance of this resistant starch is it have low glycemic index. So low glycemic index means that is a, after consumption of such food, there is a no lower increase of glucose in a blood. So high glycemic index food means those foods which give a more glucose after consumptions. Low glycemic index means that give a less glucose. So generally whatever the carbohydrate we consume from a dietary food, so finally it is converted to the glucose in our body. So these foods, according to the providing of these foods, they provide a glucose. So the concentrations of glucose provided by that foods, they are classified into two types. So one is the high glycemic index, and next one is a low glycemic index. So high glycemic index food means those food which yield a more amounts of glucose. So that means it rises a blood sugar after consumptions. And its low glycemic index foods means after consumptions of these foods, they do not increase retain, so that it prevents the this one from a this carcinoma of a intestine. So another one is that is the this cholesterol lowering effects. So these dietary fibers, they can lower cholesterol, blood cholesterol by two ways. One is it prevents the absorption of cholesterol from small intestine. That means it prevents the absorption of dietary cholesterol from small intestine. So that cholesterol will not rise in the blood. And next one, that it inhibits the absorption of bile salt. So that means it increases the excretion of bile salts from a stool, so that what happened, the bile salts will be decreased in the body, and so that the excess cholesterol will be converted. That is, this cholesterol is used for a synthesis of bile salt, so the cholesterol level will be decreased. So another one is anti-hyperglycemic effect. So that means these soluble fibers. So what they do, they reduces the time of travel of a food from stomach to small intestine. So which slow downs the absorptions of glucose from a small intestine. So that lead to the slowly rise of glucose in the blood. So which is called as an anti-hyperglycemic effect. So hyperglycemic means that is a rise blood glucose level. So with the consumptions of this dietary fibers, then what happened? There is the slowly rise in the glucose. So slowly absorptions of glucose from a small intestine, which prevents the sharp rise of glucose in the blood. So which is called as anti-hyperglycemic effects. Satiety effects. So if we consume large amounts of dietary fibers along with the another dietary macromolecules, then it's make a bulk of food, so that which makes a fullness of a stomach. So that is called as a satiety effect. So these are the advantages of dietary fiber. Disadvantage of dietary fibers. So one is that is the fibers they provide they not only inhibits the absorptions of this cholesterol and then there's a toxic metals. So this also inhibits the absorptions of certain these essential minerals like this calciums, irons, and zincs. This fiber lead to the decrease in these minerals to the body. And next one is it also decreases the intestinal protein absorptions. So some fibers are degraded by intestinal bacteria, so which lead to the productions of a gas in the intestine, so that causing a flatulence and abdominal discomfort. So the mechanisms that we have studied during a lactose intolerance. So these are the different references from which I took these notes. र अनि एउटा कुरा जुन छ के छ this कमला अधिकारी, so she asks about the solvent drag. So solvent drag भनेको चाहिँ जस्ट त्यो सेलहरू बिचको जुन स्पेस जुन हुन्छ त्यो स्पेसबाट चाहिँ के हुन्छ त त्यो सल्भेन्ट मुभ फ्रम इन्टेस्टिनल लुमेनबाट चाहिँ जुन इन्टरटेसियल स्पेस अथवा जुन जहाँनिर ट्रान्सपोर्ट हुन्छ सो त्यसलाई चाहिँ सल्भेन्ट ड्र्याग भनिन्छ। सो यो जुन इन्टेस्टिनल लुमेनबाट चाहिँ जुन इन्टरटेसियल स्पेसमा चाहिँ हाम्रो कुनै पनि सोल्युट्सहरू अथवा जुन सल्भेन्टहरू जुन हुन्छ त्यो चाहिँ ट्रान्सपोर्ट गर्नुको लागि चाहिँ दुईटा माध्यमबाट जान सक्छ। एउटा भनेको चाहिँ इपिथेलियल सेललाई पार गरेर जुन सो इपिथेलियल सेललाई पार गरेर जुन जान्छ त्यसलाई चाहिँ के भनिन्छ त द्याट इज द जुन ट्रान्स सेलुलर ट्रान्सपोर्ट भन्यो। अनि अर्को चाहिँ जुन हुन्छ द्याट इन्ट्रास जुन सेलहरू बिचको जुन स्पेस जुन हुन्छ त्यहाँबाट ट्रान्सपोर्ट हुँदा चाहिँ प्यारासेलुलर ट्रान्सपोर्ट जसलाई चाहिँ सल्भेन्ट ड्र्याग पनि भनिन्छ। सो यु क्यान सी इट इन अ फिगर जुन छ। सो दिस इज अ फिगर। सो एउटा भनेको जुन छ इन्टेस्टिनल डोमेनबाट जुन सेल भएर चाहिँ जुन इन्टरस्टिसियल स्पेसबाट चाहिँ जुन ब्लडमा जान्छ सो द्याट इज अ ट्रान्ससेलुलर एब्जर्प्सन। अनि अर्को चाहिँ यो सेलहरूको बिचमा जुन ग्याप हुन्छ सो त्यो ग्यापबाट यदि कुनै चिजहरू जुन ट्रान्सपोर्ट हुन्छ भने द्याट इज द प्यारासेलुलर एब्जर्प्सन्स ओर सल्भेन्ट ड्र्याग भनिन्छ। सो इफ नट ओके, सो आई विल प्रोभाइड दिस स्लाइड via the email address, and on next class we will learn on digestion of protein. Thank you for the class.